audience, my name is Pavel Kohout and I am PhD student at Loschmidt Laboratories, Masaryk University, Brno, Czech Republic. And today I will present to you my poster about the topic of foldability-guided sampling from protein evolutionary structured latent spaces of variational autoencoders. The evolutionary design of protein is a computational technique inspired by the natural evolution, where the organism has been optimized under environmental pressure. To study evolution, we collect and align sequences in a multiple sequence alignment, build a phylogenetic tree, and reconstruct ancestral sequences in a process known as ancestral sequence reconstruction, which can produce more stable proteins as we go against the time in the evolution. Unfortunately, ASR has limitations such as many manual steps in a tree creation and computational complexity. A way to tackle these challenges in evolutionary design might be the use of machine learning with latent space models capabilities to deduce evolutionary information from without the need of phylogenetic tree. Variational autoencoders are latent space models which consist of two parts, an encoder and decoder connected by the informational bottleneck called latent space. By sampling from this space, VAE can generate new meaningful samples and also can generate new meaningful protein sequences. The study of Ding and authors showed that latent space structure can capture phylogeny and that the evolutionary trajectory from the extant sequence to the common ancestor is strongly correlated with the latent space position, as ancestral sequences are mapped closer to the latent space origin. The authors did not try to use VAEs to generate ancestral sequences. Therefore, we decided to explore the capabilities of variational autoencoders with evolutionary data to design novel functional proteins with higher stability. We designed a pipeline to gather enzymatic data through the enzyme miner tool. We optimized VAE architecture to reconstruct sequences and to be able to capture evolutionary dependencies within the latent space structure. We then designed evolutionary inspired sampling strategy, sampling from the query embeddings to the latent space origin, and we collect multiple statistical values for generated sequences such as sequence similarity to the training dataset or model probabilities. We use these metrics as a filters for a selection of the most promising variants for biochemical characterization. We perform our study on haloalkane dehalogenases with DHA protein as a template, and we found out that single dense layer with two-dimensional latent space is sufficient enough to reconstruct MSA sequences and capture phylogeny. We did 3 rounds of experiments, and we experimentally characterized 20 designs in total. In the last third round, we observed the best stabilization up to 9 degrees. As half of our designs was insoluble, we decided to study more deeply in silico metrics for design scoring to guide selection and sampling of the sequences from the latent spaces. For this purpose, we developed a foldability metric which combines structural and inverse fold predictors. With 2 pipeline, we predicted a positional specific scoring metrics for given sequence, on which we then projected 1 hot encoded amino acids. The average of individual positional probabilities is then represented as a foldability of a given sequence. As we have referenced wild-type sequence, we define a relative foldability score as the logarithm of ratios between foldability of mutant and wild-type sequence, with a positive value meaning better solubility. We predicted foldability scores for our VAE designs with good predictive performance. When we applied the foldability score metrics to a bigger Pfizer dataset of 56 ancestral sequences from one phylogenetic tree, it performed scores from one of the best solubility predictors, SoluProt. Inspired by these promising results, we expand our VAE pipeline by the foldability-guided sampling. We predicted the foldability landscape of the latent space and designed two foldability-aware sampling strategies. The first one, mutagenesis strategy, randomly mutates the sequence on the input and embeds it to the latent space. Then, it selects mutants dominating the other mutants with the foldability score and the smallest distance to the latent space origin. This procedure is repeated until the convergence to the latent space origin. These two objectives can also be studied directly in the latent space. Therefore, we develop a second strategy when we apply the covariance matrix adaptation evolution directly on the latent space coordinates. The results demonstrate that both strategies can yield sequences with higher foldability scores than the previously used evolutionary trajectory strategy, thus potentially improving the success rate of generated variants. Thank you for your attention, and I would like to express my gratitude to the COSAM ITC grant for providing me financial resources to attend this conference.